I'm going to tell you about machine learning models for single cell and regulatory genomics. So I'm going to just start with a little bit of background and motivation so that we're all on the same page. And I hope that people have this view in their head of how transcriptional gene regulation works, that there are transcription factors that bind distinguished elements in the genome, promoter regions, but also more distal enhancer regions. But these distal enhancers are looping around in 3D and making contact with the promoter. And this enhances transcription of the gene, okay, and upregulation of the gene. All right, and what's happened in the past few years is we have excellent data on 1D epigenomic data, increasingly at single cell resolution, as well as 3D genomic data that tells us about the connectivity of the genome in 3D. Okay, and this leads us to new problems, new machine learning problems that we can solve to try to understand this data. Okay, why do we care about gene regulation? So I know people are from many different backgrounds, and the scope of this workshop is wide. So in terms of motivation for studying gene regulation, beyond the basic, fundamental importance, for population genetics, for human genetics, the important hypothesis is that a lot of the non-coding genetic variation that we see that is associated with disease is actually affecting these regulatory elements and therefore changing regulation of gene expression. All right, and so to understand genetic variants associated with disease, we need to understand how genes are regulated. Okay, so that's one motivation. Another motivation is that in disease, certainly in cancer, absolutely in metastasis, what you're seeing is an aberrant differentiation process. Okay, so cells are in different differentiation states, and you want to study that not just at the phenotypic expression level, but also mapping these events, these enhancer and promoter events, and understanding the transcription factors that are driving differentiation states, okay? And third, cell engineering, okay, both in the CAR T cell space and also in cell replacement therapy. We need to push cells into different states, and we do that by understanding transcription factors, their targets, how to achieve certain differentiation states. All right, so largely I'm going to talk about chromatin accessibility, ways that you map the accessible elements, regions that are held accessible, that become accessible because there are DNA binding proteins there, and also talk a bit about 3D genomics. All right, so a bit more background. Okay, so if you indulge the biology for a bit. Okay, I just want to make sure people have seen this kind of data and understand the relationship between chromatin accessibility and gene expression. All right, so I could show many papers from many people. This is one of my papers. That was one of the first cancer immunology papers that my group did. And it was looking at what happens to tumor-specific T cells as they fail to control the tumor and they progress to a state called exhaustion or dysfunction, okay? And what we're looking at here is the locus that encodes PD-1 in the mouse. So PD-1 is an immunotherapy target, and also the locus that encodes interferon gamma. And what we're seeing is these are CD8 T cells as they, sorry, I keep losing my cursor. Okay, as they're progressing towards dysfunction, okay? And at the top are, the normal differentiation states naive effector memory that you have in a functional immune response, okay? And if you compare functional to dysfunctional, you can see at these loci and genome-wide, there are changes in chromatin accessibility, okay? There are events that, for example, are specific. You only see in the dysfunctional state, and you don't see in functional cells. There are events that are supposed to be big that become small. So both on-off and quantitative changes, okay? And genome-wide, you can see that the chromatin accessibility is changing and defining these different differentiation states, okay? Okay, so that was some years ago. And then you fast forward to about 2021, we can do this all in single cell, okay? So now we're looking at functional and dysfunctional immune responses, again, T cells, CD8 T cells, at single cell resolution, okay? And what I'm doing here is I'm showing you pseudobulk data, so I'm clustering single cells, and then I'm looking in pink are the dysfunctional immune response. It's a chronic viral infection model. And you can see these events that we saw in bulk at the single cell level, okay? So including this dysfunction-specific enhancer of PD-1, all right? So quite exciting that we can see these events at single cell. More is true, okay, that we can also these days map at high quality the 3D interactivity of the genome, okay? The single cell data is coming. I'm only going to talk about bulk, high C, and high CAR. So high CAR is a variant of high C that uses the TN5 transposase like ATAC, okay? So it kind of gives you also an ATAC readout for free. And what we're looking at here is part of the 3D contact matrix where, okay, so I have to look over here. I'm circling these events that are telling you about contacts, interactivity, interactions between enhancers and a promoter of a developmentally important gene, PD1 in, PDX1 in pancreatic differentiation, okay? So from the top to the bottom, embryonic stem cells, definitive endoderm, and pancreatic progenitor 1. This is a guided differentiation system. And what we can see now with this 3D genomic data is we can see the gain, okay, I again have to look over here, the gain of these interactions between enhancers and the promoter of this developmentally important transcription factor, actually. Okay? So we have this really nice quality data. We can relate 3D connectivity and also accessibility changes potentially to gene expression changes, okay? All right, so what I'm gonna talk about, I'm gonna talk about methods for single cell attack, methods for predicting the high C contact matrix, and also methods for inferring gene regulatory models from single cell multi-ome data. So first off, I'm going to tell you, First off, I'm going to tell you about a sequence-informed embedding model for single cell chromatin accessibility data called CellSpace. Just got published a few months ago. I'm next going to tell you how you can predict the contact matrix, the high C contact matrix, from single cell attack alone, okay, using a supervised deep learning model. And finally, I'm going to show you fairly simple model that we train on single cell multi-ome data to be able to learn the cell type specific enhancers of genes. Okay, so I hope to get through all of that in the 40 minutes that I have. Okay, let's start with CellSpace. All right, so single cell attack, is a technology that's really come of age. If you have the money, you should always do single cell attack instead of bulk, I think, so because you learn more. and it allows you to map chromatin-accessible events, which we think of enhancers, promoters, maybe other structural elements like CTCF binding sites, okay, in single cells. We can also infer transcription factor motif accessibility in single cells. We can find different cell types and differentiation states in a complex sample. Okay, the thing is that it is very sparse and challenging data, high dimensional, and most methods for analyzing this data just mimic the single-cell RNA-seq pipeline that you've presumably already seen. So instead of a cells by genes matrix, you start with maybe a cells by peaks matrix or cells by variable genomic tiles. You do dimensionality reduction, you do nearest neighbor graph, you do graph-based clustering, and visualization by embedding. Okay, and that's what's happening in the, for this data, which is from Buenrostro et al. It's bone marrow data. You can see, you know, if you pseudobulk it, this bone marrow data looks like bulk ATAC, but you can see the events in individual cells. Okay, and it is very sparse, right? The dynamic range is 0, 1, or 2, because in a diploid genome you have 2 chances of seeing the event. And if you do a standard kind of analysis, you get something that looks like there's structure, looks like everything is working, but there's a problem, okay? And the problem is these 2 green cell types, okay? They're hematopoietic stem cells from 2 different donors, all right? So instead of clustering by the cell types in the bone marrow and seeing these different states in hematopoiesis, it's all confounded by a donor batch, all right? And, you know, sometimes I present this and people are, you know, what about individual differences between my hematopoietic stem cells and yours? Yes, absolutely, there are individual differences. There are events that might occur because of genetic variation in one sample and not the other, but it's not reasonable that the cells of the same type between 2 donors are as far apart as different cell types in the same donor, right? It's not reasonable. This is a batch effect. Okay, so what do we do? So what we thought to do was use the fact that we have additional information, sorry, in this system. Maybe this is not, you can hear me? Okay, I won't touch my mic then. We have additional information. So underneath chromatin accessible events, whether you do peaks or whether you have tiles, just, you know, 500 base pair tiles, there's DNA sequence, and that's the important information, right? So that's where there are binding signals for transcription factors. Transcription factor binding defines the cell type. And maybe that sequence information is more conserved between batches than the actual events that are captured. All right, so what we do is we learn an embedding of both cells and DNA k-mers into the same latent space, okay? The k-mers we use are 8-mers for convenience, okay? And we generate training examples on the fly. So we have our chromatin accessible event. We sample a fixed length sequence and turn it into k-mers. That sequence was accessible in, you know, one cell that we sample, not accessible in some other negative cells that we sample. Okay, so we have a sampled positive cell, a sampled set of negative cells, and we're embedding k-mers and cells into the latent space, pushing k-mers towards the cell in which they were open and away from cells in which they were closed. Okay, so every event, sample a sequence, it generates a training example, and we push cells and k-mers around. At the end of the day, all the k-mers and all the cells are embedded in the space, and now from that embedding, you can do clustering, you can do your UMAP visualizations. Note, we are not explicitly representing the peaks or tiles themselves, and we're not explicitly representing transcription factor binding motifs, okay? But after the fact, we can embed peaks, we can embed motifs into the space and then do things with that information. Okay, everyone with me? All right, so I'm just going to, you know, for people who prefer the math to the verbal description, this algorithm is from CellSpace. It was originally designed by Facebook AI to embed tweets and hashtags, right, into the same latent space. And you have a word embedding, right? So you can imagine the analogy. We get training examples. The left-hand side is a bunch of k-mers. The right-hand side is a cell, right? And the k-mers and the cells are tokens in the system. We're learning a coordinate, right? We're learning an embedding for all our tokens. And we're trying to push the left-hand side, the similarity between the left-hand side and right-hand side for the true positive examples closer than the negative examples by a margin, okay? So, and I'm simplifying a lot of things. We're not just using a bag of k-mers approach. There's actually an n-gram approach where k-mers that are close to each other also feature in the representation. And there are other details to make it very scalable, okay? But the basic idea that you're doing this margin-based loss over positive and negative examples, you know, that's the basic idea. Okay, so let's go back to that bone marrow data set. I should point out we have ground truth labels because they sorted these cells by flow, so we know the true identities of the cells, not always the case. And just to remind you, this is the standard embedding, right, which is sort of confounded by batch. And this is the CellSpace embedding, where we're not doing anything. We're not, you know, explicitly correcting for batch. We're just letting the DNA k-mers inform the embedding of cells, okay? And what happens is that all the hematopoietic stem cells in green from both donors embed together close to each other in the latent space, which you can see in this UMAP. And also, it seems like the latent space is capturing the correct developmental hierarchy of hematopoiesis because, you know, if you, look up here, so this is the kind of hematopoietic tree, you can see that that tree is kind of captured in the UMAP. That's the UMAP of the CellSpace embedding, okay? In fact, you can even do a trajectory inference method like Palantir, okay? So start at one of the hematopoietic stem cells and just find all the most differentiated cell types correctly and the differentiation paths towards those cell types, okay? So somehow, by using DNA k-mers to inform the embedding, we're getting a bit better latent structure and mitigating batch effects. Okay, one more thing is true. You can take transcription factor binding motifs, right? So their position weight matrices, and you can turn it into a set of k-mers, right? So one way is just to get the consensus motif and tile with k-mers, embed in the space, and now I can compute a proximity between transcription factor binding motifs and cells I can put it in the whole nearest neighbor graph. And what you're seeing, actually, if you see closely here, I know it's a little hard to see, these labels are actually the embedded motifs. And what you can see is that the motifs are embedded close to the cells in which they are accessible. Okay, so for example, if I can find my HOXA9 is a known hematopoietic stem cell transcription factor. It's embedded near the green cells. Okay. Okay. So in review, people wanted to show that we could do de novo motif discovery. So we have all the cells and all the DNA chimeras embedded in the same space. Can you find new motifs? Okay. So I don't think we showed that you can find new motifs, but we showed that you could find known motifs de novo. Okay, so what we did was we took 10 mers, embedded them based on the 8-mer embedding, and then for every cell cluster, we found the 10 mers that were close. We clustered them by their sequence content and produced motifs. And you actually get all the correct motifs associated with all these cell types. And it's too hard to see, but that's what's shown in this rather busy diagram. And I'm sure you could do more. Okay, so there really is transcription factor signal information in the embedding of the chimeras. Okay, one more thing we can do. We can take important transcription factors. If we know they're binding motifs, we can embed the motifs. We can compute the similarity between motifs and cells in the latent space. And we get a score, a transcription factor activity score. And that tells us where the model is saying certain transcription factors are active. Okay, so I'm doing that at the top of the slide for CellSpace on a human cortex data set. And this data set is actually a multi-ome data set where we have single-cell ATAC and single-cell RNA from the same cells. And for example, so in the middle is the expression for different transcription factors. And here are the motif-based transcription factor activity scores. Okay? And you see that the expression and the activity are fairly correlated. So for example, ATAC-seq is only expressed in radial glia, and we're saying it's only active based on the motif embedding in the radial glia. Okay, at the bottom of the slide, I'm comparing to another model. It's a very nice model. It's based on the Bassett deep learning model for sequence, and this is called single-cell Bassett. It also gives you a sequence-informed embedding. And this is just we're using deep sequence models as well. So it's just a cautionary note that sometimes the sequence information that you're capturing can be hit or miss. All right, so SC Bassett has a nice way of, you can do a motif injection and predict where that motif would be accessible across your single-cell ATAC atlas. And you can see for ATAC-seq, it's just wrong, right? It's just, it's not active in the right place. But for some of the other transcription factors, it's doing better, all right? So, the deep learning models can correctly or incorrectly learn sequence information. They can also overfit, all right? All right, I just want to show a little bit more and then move on. Okay, another example, since we are often interested in clinical samples. Clinical samples have batch effects. This data is from a study from Satpathy et al. They did single-cell ATAC in bone marrow and peripheral blood, multiple donors, big atlas. And in order to get the embedding that you see on the left, they had to do a kind of bespoke procedure. They're using standard methods, but they are kind of doing a custom batch correction. So embedding, batch correct, re-embed, and then they labeled their data. Okay, so that's this sort of custom approach. And On the right is CellSpace, no batch correction. Okay, we're not even using a batch covariate. Okay, so we're just embedding all the data, and the structure is reasonable, right? We're clustering the cell types properly, and we're also finding correct developmental relationships between cell types. So the bone marrow cell types are here. You have peripheral B cells, and then you have T cells and NK cells, the sort of lymphocytes, T lymphocytes. Okay, final slide on this, and also cautionary tale about batch correction. So, these batch mitigation properties of CellSpace, I think it is important because batch correction can fail, and you don't know if it's failed unless you already know the answer. Okay, so going back to this study that we did, two groups, John Wary and Satpathy lab, again did this kind of functional versus dysfunctional immune response, looking at CD8 T cells, so similar design in two studies, and they did single cell attack. Okay, and at the top we see the CellSpace joint embedding. Okay, here, and everything makes sense. The naive cells are here. You have a exhausted progenitor population, and a progression to terminally exhausted, and there's also another destination, which both papers describe as a natural NK-like exhausted state. Okay, so at the bottom is a very widely used and good batch correction method called Harmony. We've used it too. It can work, right? But this is an example where it's failed. All right, so you apply Harmony, two data sets, very similar data cell types and developmental design or experimental design, and it's just turning all the data into a blob. Okay, and in particular, you can see that the naive cells from the two studies are separated. The terminally exhausted cells are in two places, right? So this is a fail, all right? And often, right, you're in a situation where you don't know whether the batch correction is overcorrecting or undercorrecting or working, right? So I think at least this gives you another source of information, the DNA k-mers, to guide the embedding and mitigate batch effects. Okay, so that's the first thing I wanted to tell you. CellSpace, which is a DNA k-mer-based embedding that learns latent structure for single cell attack. It's very scalable. I didn't show you that. It can scale to 1 million cells. You can see that in the paper. You can also embed transcription factor binding motifs and get motif activity scores based on proximity in the latent space. It mitigates batch effects, and often you don't even need, every example I showed you, you don't need to correct for batch effects explicitly. I didn't show you this, but you can even take data sets that are processed separately by different groups and with respect to a different peak atlas, and you can still integrate. And there we use the batch covariate just to make sure that when we sample positive and negative cells, it's always from the same batch. So we don't want to push the batches apart. So that's in the paper. Okay? All right, so that's the first thing I wanted to tell you about. Okay, the second model, and I see I'm not going fast enough. Okay, the second model I can show you quickly is predicting the contact matrix from single cell attack. Okay, what is the contact matrix? So there are a bunch of 3D genomics technologies. There's Hi-C. I showed you some HiCAR data. But basically, what you're doing is capturing contacts by a workflow that looks like this. You're cross-linking the DNA in situ in the nucleus. You digest usually with a restriction enzyme cocktail, and then you're ligating the ends and pulling down at the end of the day, you're going to have a paired-end sequencing libraries of these chimeric fragments, the gray and blue, right? So the gray and blue parts were maybe far in the linear genome, but they were close enough in 3D to be captured by this proximity ligation, right? And what we get from these paired-end reads, we call the read pair a contact, right? We map, usually we bin the genome and we map what are called the anchors, right, to bin I and bin J, and that's, we increment a contact in the 2D matrix, contact IJ, okay? So Cij is a number of paired-end reads with anchors in bin I and bin J. We get something like this. I'm taking the matrix, putting it on its side, and the only thing you need to know is that usually what people do is they normalize by a matrix rebalancing, and the main thing you see are these triangle structures called TADs, topologically associating domains, okay? And the picture, right, though you shouldn't believe these cartoons too much, right? Everything is dynamic, everything has an on-off rate, but you have dynamic loops, and people believe the loops extrude out of a cohesion ring and are anchored by CTCF, okay? And that's what you're seeing with these big TADs. We have a statistical procedure where, using negative binomial regression, we estimate a background model. It depends on genomic distance and other systematic sources of variation associated with the bins, and we can estimate a p-value or binomial z-score of, how strong is that contact relative to our background model. And that's what I'm showing here. And you see with these high CDC z-scores, and we can also draw loops, we're getting more information, including enhancer promoter information, not just this kind of big triangle structures. Okay, so that's all you need to know about how we're processing the data. Okay, can we predict the contact matrix from single cell attack? All right, so we're inspired by this older method called Cicero. It's very popular. You'll see people using Cicero quite a bit. And their idea is that if you have peaks that are co-accessible, they open and close together across single cells or across metacells, then maybe they're more likely to have an interaction in 3D between them, okay? And just to see, is there support for this? What I'm showing in the corner is the co-accessibility matrix, similar to what Cicero uses at the top, and the actual high-C contact matrix normalized the way we normalize with high CDC scores, okay? And you can see the co-accessibility matrix has some information about 3D organization, but it's also very noisy, and there's this plaid structure, and an unsupervised approach that just takes this co-accessibility matrix and does some statistical processing is going to have low accuracy, okay? Here's another way of thinking about it. Here's the distribution of co-accessibility in orange for the true interacting pairs, and in blue for all pairs, right? And there's a little shift, right? So that if you have a 3D interaction, you're a bit more likely to be co-accessible, okay? So this is a subtle signal, but one way to make use of subtle signals is through deep learning, okay? So this is a model called ChromaFold. We're predicting the 3D contact matrix by supervising. It's a convolutional neural network. It's a fairly lightweight model. We use 3 sources of information. So from the single cell attack, we get a pseudobulk signal. Okay, so that's here. We need a bit of sequence information. We use CTCF motifs, so just a motif track, because those CTCF motifs are associated with TAD structure, and that's useful. And then we also use the co-accessibility matrix over metacells. This is very similar to what Cicero did. Okay, so we have 2 feature extractors, one for the 1D, one for the co-accessibility. In here is an outer concatenation of the 1D signal. So we're turning it into kind of an L by C into an L squared by 2C input. And then we're just predicting along this V stripe, and we glue the V stripes together. Okay. All right. How does this work? Okay, so the first sanity check is if you train on some chromosomes and hold out other chromosomes in the same cell type, how well can it work? Okay, so what we're seeing is the ground truth here and the prediction here. Okay, and this is CTCF motif. This is CTCF chip-seq, which we don't use. But just so you see it, the ATAC pseudobulk and the co-accessibility, which you see is very noisy. Okay, and if you look at this, you see this signal is learnable. Okay, so this accuracy is pretty good and pretty usable. All right, so we don't want to just predict in the same cell type. We want to learn a model that generalizes to new cell types. So that's what we're doing here. So now this is the ground truth. This is the model's prediction. And I'm showing what happens if you take away the CTCF motif. And then you don't learn the structures as well in a new cell type. Okay, so, but this model versus this, pretty good. Okay, so the accuracy is usable. Okay, you can also go to a new genome. All right, so we take these models that are trained on human cell line data, and we predict in mouse. And we happen to have very wonderful collaborators who are generating data that is very difficult to generate and is high quality. Okay, so here, what I'm showing you is the ground truth high C in germinal center B cells in the mouse at the BCL6 locus, and that's from the Ari Melnick's lab and from Sasha Rudensky's lab, regulatory T cells at the IKZF2 locus. Okay, and this is the prediction. All right, so the prediction is actually good enough to look at, find the significant interactions or the highest scoring interactions and make inferences about what enhancers might be interacting with the promoter. Okay. I think I have to go quickly, so I'm just going to say, we did a lot of benchmarking. It's in the bioRxiv paper. This isn't even fair, right? So we're comparing against Cicero, which is an unsupervised method that just takes the co-accessibility matrix and does a graphical lasso. It doesn't perform very well, so this is R loops, and this is the Cicero. Oh, so experimental, ChromaFold, and the Cicero. Okay, and you can see if you look at predictions based on significant interactions that we call from ground truth, it doesn't do very well. Okay, which is not surprising. It's a hard problem. Last use. Okay, and maybe some people saw this coming. If you have single cell ATAC and high C in a complex tissue, you can take the ChromaFold model, fine-tune on that data, and then make predictions from ChromaFold for each cell type. And that's like a deconvolution of the bulk high C. Okay? And here we're doing this in pancreatic islets. So there's published bulk high C data, there's single cell ATAC, we fine tune, and then we're making predictions in beta cells and alpha cells. For a long time, we didn't know how to validate this, and then someone actually went to the work of sorting out alpha cells and beta cells and published high C, and then we could benchmark. Okay, and it's, okay, not perfect, but let me just show you. Okay, so this is the glucagon locus. Alpha cells make glucagon, beta cells make insulin, right? And this is the insulin locus. And what you can see in the bulk, right, you see this structure, and that structure is coming from the alpha cells. This is the ground truth. I mean, at least we're getting this one very clearly, right? This loop, this is the prediction, okay? And so that's our prediction in alpha cells and prediction in beta cells. So we're getting certain interactions correct that you can see in the ground truth. It's not perfect, okay? All right, so I think I have about 6 minutes left to tell you one more model. And I think this happened last year that I ran out of time at exactly the model that might be the most useful to the population genetics people. But this is now published, okay? So this came out in Nature Genetics earlier this year. It's very simple, okay? So I can tell you very quickly what we do. If you have multi-ome data, so you have single-cell ATAC and single-cell RNA from the same cells, we learn a gene-level single-cell model where we're predicting the RNA expression of a gene from the single-cell accessibility. Okay, so so the prediction, the training examples are single cells. We're focusing on a single gene. What we do, so we use the gene locus as well as 250 kb upstream and downstream of the gene by default. And we're just using 500 base pair tiles, no peak calling, okay? And we are literally, doing regularized Poisson regression, predicting expression from chromatin accessibility. You have to do a few things in terms of normalization correctly for all of this to work, but it's very simple, okay? And at the end of the day, if I can find my cursor, you get regression coefficients, and you can then take the model and do Shapley value analysis per cell type to find cell type specific enhancers. Okay, so let me tell you that quickly and not go over 1205. I think it's easiest to see with an example. Okay, so this is the model for ZEB2. It's the gene here. It's transcribed, right to left. Okay, and what I'm showing is so this is just PBMC data. This is peripheral blood, multi-ome. It's on the 10x website. Okay. And I'm showing you the pseudobulk of the ATAC and also the distribution of gene expression per cell type. And I'm showing you here the regression model, okay? So what is the regression model doing? You can see that if the coefficients are a bit higher all over the locus, okay? That makes sense, right? The promoter and the accessibility of the locus tells you something about how robustly the gene will be expressed. But it's also finding other events, okay? So it's finding these distal events, right? It's saying these sites are important. And indeed, whenever you see these events accessible, the expression is shifting to higher, okay? But it's not just finding highly accessible events, because you can see over here, there's this event that's highly accessible, and the model puts a regression coefficient close to 0. And if you look at the gene models, you can see, oh, well, this is the promoter of another gene, and it doesn't matter for explaining the expression of ZEB2, okay? So it's just a very simple way of learning a model where you're taking all the information at once, right? Not pairwise correlation, but a regression model. And you're getting a model of how that gene is regulated. What are the accessibility events that are important? You can also see that the model is constraining all the coefficients to be positive. That's actually important for interpretability, so the model can't hedge between positive and negative examples. Okay. This is just a sanity check that the SCAR-linked predicted expression is better than all the other ways that people try to turn ATAC-seq into an RNA-like approach. Archer gene score, Cracker, Dork score, and not surprising because it's supervised. Okay, so this is on hell.cells, but one more thing to tell you, and then I think I should go to the end. So what we can do, so we have this model, regression model. This is another example. We're modeling CCR7, again in the PBMC. Okay, CCR7 is this little gene here, and we're looking at the context. We can take the model and then do a Shapley or approximate Shapley analysis, so just feature attribution per cell type, which tiles are important for predicting gene expression in, for cells, looking at cells in one cell type. And we can take those Shapley scores and assess their significance, and that's what these blue dots are. And then if you have a fine map disease-associated variant, you can see if your variant falls in a tile that has a high Shapley score and which cell type it's important for. Okay, so this is an asthma-associated variant, and you can see that it's significant just in the CD4 and CD8 T cells. And it makes sense because this gene, CCR7, is important for T lymphocyte homing to the lymph nodes. Okay, so it's a way, if you have multi-ome data, of understanding how the genes are regulated, but also annotating non-coding genetic variants and associating them, with the gene and with the cell type where they're active. Okay, and I'm not going to talk about chromatin potential, but just go to the end. Okay, so let me thank all the people involved. Thank you for your attention. If there's time, I'm happy to take questions.